In this video, I'm going to be talking about the process of DNA replication. The aim of DNA replication is to produce two completely identical DNA strands. Due to the complementary nature of DNA, where one strand codes for the other strand, you're able to use each strand as a template for a new strand. So the first thing that needs to happen is that the strands need to be separated, and that is done by an enzyme called DNA helicase, which works its way up the DNA helix, unzipping it and unwinding it, to reveal what we call a replication fork containing single-stranded DNA. At this stage, another enzyme called DNA polymerase comes in and starts to draw in DNA nucleotides, which are complementary to the DNA nucleotides on the single-stranded DNA. As it moves up, a DNA strand will form behind it, like this. So we have A here, so that would be T, and then A, and then G, and then C, and then A, and then T. And you will see that the DNA polymerase is able to work continuously along this strand. Consequently, we call this strand the leading strand, and that allows the DNA polymerase to work continuously. On the other strand, the DNA polymerase has to work in the opposite direction, because you'll remember that DNA itself is anti-parallel. So if it can work in this direction on this strand, it has to work in this direction on this strand, which inevitably means it has to work in small fragments as new sections of the replication fork are exposed. So it would start up here, and then it would start to work down the strand like this, where it would fall off and then go and join up here, and so on and so forth. So as it moves along, it also brings in the complementary bases. So ATC, ATG, C, T, A, and then those are joined together. So this strand we call the lagging strand, and it results in the formation of Okazaki fragments, which are these small segments of the DNA which is replicated on this strand in a discontinuous fashion. Those fragments are then stuck together, and you end up with two completely genetically identical DNA strands. We say that DNA replication is semi-conservative. The reason for this is that, as you can see from this diagram, the original DNA strands, shown in black, are retained in each of the new molecules. So this was the original strand, it's separated, and now in our new strands, in our new DNA molecule, it's made up for one old strand and one new strand. And that pattern continues as replication continues. And consequently, we call this semi-conservative replication. And it was originally proposed by James Watson and Francis Crick shortly after they had established the structure of DNA.